Okay, good morning, everyone. This is EMX, first Monday of the month, 8 a.m. Pacific time. And we've got two exciting speakers whose theme is Cryo-EM, I believe, today. So this will be, it's the end of our academic year, so we will take a break for summer. So this will be the last one of the current series. Seems like we only started a few weeks ago, but in fact, it was a few months ago last year. And hopefully we have a summer of recovery and rejuvenation, and we will start again. Unfortunately, the first Monday of September is Labor Day in the United States, and so we will avoid that. And we'll start again the first Monday of October, and we'll send out plenty of notices ahead of time. And we will rejoin. But hopefully we'll be back in the office. We see that one of our speakers already is. Thank you, Lena. But at Stanford, we are still sheltering in place, as it were. I'd like to thank Ziwen, Raphael, and Yusong, who've helped really organize this EMX symposium. It's been a lot of work for everyone, and they've done a fantastic job. But without much more ado, then I will remind everyone about the summer break at the end. Very pleased to, before passing over to Yi to introduce our first speaker, Chiu, say a few personal words about this. Came to Stanford 4 years ago from Baylor, very successful career there, and has had a huge impact. But Wah and I go back to the old days in the 70s when Wah was a graduate student with Bob Glazer at Berkeley, and I was a young postdoc in Gareth Thomas's group. I used to go over, and Bob Glazer had an optical bench set up, and it was quite nice. And so to analyze, of course, high resolution images, and we didn't have that, and they were very gracious in offering that facility open to us. So I came over fairly regularly to take advantage of that. Of course, everything is done so much faster now on a computer with a fast Fourier transform, but in those days, and it was good for me to interact with Wah and see what they were doing there, pioneering work in the Bob Glazer group. So I will pass over to Professor Yi Che, who will do the formal introduction. Yi.
Thank you, Bob. Yeah, I'm Yi Che, a professor in material science at Stanford right here. It's been a great fun co-organize this with Bob, Wah, and Jen to bring all the electron microscopists together around the world. Now today is a great pleasure to introduce Wah. Bob already mentioned a little bit about Wah's background. Well, let me mention a few of my personal words as well. Several years ago, close to 5 years now, about 5 years, the reason I got into cryo-EM research, Wah, is you are one of the main reasons I got into cryo-EM. At Stanford right here, we are so fortunate to have you joining several years ago, leading up the effort in cryo-EM electron microscopy. For the audience right here, you all know Wah is a world-leading person in the cryo-electron microscopies. I just like to mention a few of his great honors, awards he received. He's the member of U.S. National Academy of Science. He's also a member of Academia Sinica, Taiwan. Back in 2014, he received the Distinguished Science Award from the Microscopy Society of America. Well, Wah is amazing collaborators during COVID-19. Well, it turned out to be he and I worked together quite a bit on figuring out, the facial mass, the filtration, study how the particle distributed, how the virus interacts with the facial mass. Through this first-hand experience, I just learned tremendously from Wah. without further ado, let me pass this on to Va.
Okay, thank you to Bob and Yi for these very kind introductions. It's my great pleasure this morning to share with you some of work that we have been doing at Stanford and my perspective about what Cryo-EM can do beyond the ordinary. First, I'd like to share with you about the statistics of the global activity in cryo-EM research in the last 10 years. So this is a distribution of the cryo-EM structure, which had been deposited in the cryo-EM data bank and also the protein data bank. So the purple colors are the density map, and then the green color are the structure that have the accompanying coordinates. As you can see, there has been exponential growth in the last few years. So it's something like 100 maps that are being produced globally. Among all these structures, we look at the resolution distribution shown in this histogram here, that in the last few years, there are immense growth in this called near atomic resolution cryo-EM map, particularly at a 3 or 4 angstrom resolution. And this is a really comfortable resolution to learn a lot about the tertiary structure of macromolecules. So what I want to do in the next few minutes to share with you what kinds of information we can potentially get from these kinds of data. And I just want to show one slide about the activity that we, in one of our Titan Krios microscopes set up at SLAC campus of Stanford, which equipped with a K2 camera and bioquantum operated 300 kilovolt. And using one instrument in a kind of year and a half, we have been able to solve many structures, including protein machine, viruses and virus with antibodies attached to it, and also with membrane proteins, RNA protein complex, and RNA. So these are all at the range of better than 2 angstroms up to around 4 angstrom resolutions. Now, last year during the pandemics, we have been able to add more instruments due to the generous support from NIH. So in one of the instruments that we set up, again, is a Krios microscope, but with a Falcon 4 camera. So using this new camera and this new microscope last June, exactly a year ago, we were able, within 20 hours, to collect over 600,000 particles. And from there, we were able to generate a density map of this macromolecular complex called apoferritin with a total molecular weight of 480 kilodaltons. So each color represents a single polypeptide. As you can see in this density map, the shape of the aromatic residues and also the side chains of the amino acids are clearly delineated from this density map. So now here in this particular view, looking at a 4-fold axis. And on the right is the one of the alpha helix built from this polypeptide chain that we are seeing not only the clearest side chain density matched with the model. In addition, we are seeing a lot of water molecule in red and also some ions which interpret as a magnesiums. And so now in the bottom, I show 3 examples of 3 different amino acids. You can see the side chain density so quite clearly on the oxygen atoms and then also the nitrogens and the methyl side chain. So I think this has a resolution 1.27. Again, this data collected only with 20 hours. And now in last summer, we were able to install 3 instruments. So this is the result from one instrument. We actually have another instrument with the more traditional K3 camera with the bioquantum. So these are the resolution curve to show the data collected with the other microscopes. So they are ranging between 1.27 and 1.36 angstrom, which is to my opinion, are truly atomic resolutions. So while we were doing those experiments, we heard the news that in Europe, there are two groups, one in Max Planck, headed by Holger Stadt, another Saul Scheer, had at the RMB in Cambridge. They, in the case of Holger Stadt, he used a Titan microscope with a aberration correctors. And in the case of Saul's, he used the Titan with a cold field machine gun and also an energy filter made by Thermo Fisher. And they report around 1.15 and 1.22. In our case, we actually free set the data between 1.27 and 1.36. So here are the 4 amino acid example I pick up from all the structure. The column on the left are from the X-ray structure, which I consider to be gold standard. And so you can see the resolvability of each of these heavy atom carbon, nitrogen, oxygens are quite well across all these different microscopes and different settings. Now, in addition to looking at the density, we actually quantify these in terms of the resolvability. So we actually have numerical score that can rank the resolvability of different density maps and models. And so the important aspects about this is this almost identical density map we're able to create in 3 different labs in 3 countries. So that suggests that Cryo-EM is definitely a reproducible technology at atomic resolutions. Now, this atomic resolution is important because they provide information that we can begin to understand the chemistry and also the chemical mechanism of activity of the molecule. And that's also become a highly desirable resolution for pharmaceutical company to use this technique for design drugs. So I think we can declare Cryo-EM is now a truly atomic resolution for structural biology. Okay, so now in addition to protein, there's another class of important biomolecule called RNA. And there are many macromolecule has RNA as a component with proteins, but some of them has RNA alone. So far, the technology that used to determine structure of RNA containing molecule or RNA are X-ray crystallography. These are in the last 20 years. Most of these RNA structure or near atomic or atomic resolution structure were done by X-ray crystallography. But 2 years ago, all of a sudden, the Cryo-EM had picking up since 2019. It became the dominant technology. Like last year, most of the majority of structures are done by Cryo-EM. But I want to point out most of these molecules are not RNA alone molecule. In fact, they are RNA associated with the protein like spisosome and ribosome. They're different molecular machines. So I raised the question then at that time 3 years ago that why can't we use the Cryo-EM to study the RNA alone? Now the answer by the community is, oh, the RNA is usually very flexible unless you crystallize it to rigidify it, and then or you modify it in such a way that you make it rigid. So I decided with Viju Das on campus in biochemistry department, we look at this ribozyme, and ribozyme is an RNA molecule that has a function like an enzyme. And Tom Sack got the Nobel Prize in discovering these functions, and it was discovered 40 years ago. So far, this amazing RNA enzyme can undergo a series of biochemical reactions. It can cleave itself, and then it has a variety of gene regulations and translation control in the biological process. So far, there's no structures of entire ribozyme. So as a result, what the biochemist does is based on the chemical information to map out what are the likely base pairing, hydrogen bonding between these 4-letter alphabets, making up the RNA, what are they pairing? So these are completely 2-dimensional information. So we have no idea how this basic components, these nucleotides are distributed in three-dimensional space. So our approach is to actually put in the electron microscope, look at the whole intact molecule of this ribozyme, 120 kilodalton, about 3.5 times smaller than the previous case I saw. So here is the example of the images. And on the right is we call class averages by computer processing. We're able to sort out the particle of the same orientation. So these are a few classes so extracted from images like on the left. And so to my pleasant surprise, we were able to generate a three-dimensional density map, which has a 3.1 angstrom resolutions. I color it in different color because through the years, the biochemists of this ribozyme community have designated different domain based on the 2D diagram, what they are. So now the next slide, I want to show you a movie of our structure in 3D. So this is the density map of this 128 kilodaltons goes to 400 nucleotide long RNA. So I put the one on the left is the 2D diagram. And so from our map, and the model we build, it's clear that it's this long, there's a stretch of polynucleotide form a strand that a helix, and then make a 90-degree turn, and then form another helix. So then wrapping inside between these two perimeter helix is where the catalytic domains are of this enzyme. So we are able now mapping out each of these domains initially a year ago only in 2D. Now we're able to map that out in 3D. Now, in the remaining part of this movie, I want to zoom in about the detail, what you can see in terms of the molecular interaction. So in here, I'm mapping out these three domains, P4 domain, P14, and P2. In sequence-wise, they're really far apart, but spatially, they are actually quite close together, forming hydrogen bond distances. And then, so in here is quite, I zoom in a few of these nucleotide units, and the spin correspond to the hydrogen bond interaction is around 3 angstrom resolution. And in this particular script of the movie shows in this few nucleotide, they forming so-called Watson and Crick hydrogen bond and non-Watson and Crick hydrogen bond interactions. And so this is very clearly seen. And then now in this part of the movie, I'm showing this other domain on the other sides of the molecule. Again, there's a lot of hydrogen bond interaction between the bases. So amazing, this one of these molecules, which I'll show you, this very purine-rich nucleotide, this C266, is this nucleotide here making hydrogen bond interaction across in the other nucleotide in this strand, and then also make interactions hydrogen bonding with the nucleotide in these P9.1 regions. And so again, these are Watson and Crick and non-Watson and Crick interactions. Now, one of the interesting aspects about this enzyme is the magnesium were implicated to be an important components in the catalytic activity. So in some of the previous study for the crystal structure of domains of these, none of them were able to delineate clearly about the metal involved in the catalytic site. So we are actually able to pick up these three metal ion quite clearly. As a whole, for the entire enzyme, we were able to pick up 31 of these metal ion at 3.1 angstrom resolutions. So I think this basically conclude this example. Now Cryo-EM has been able to determine a structures which has been a mystery in the last 4 decades, and now we clearly delineate all the base positions and all the interaction to keep this enzyme stable. Now, this open up a new way of using the cryo-EM, looking not only the structure, but many other chemical intermediate that I delineate in the first slide. Okay, so now I'd like to move to the next example, which during the pandemic, most of the lab were locked down at the beginning, but we were allowed to work on coronavirus projects. So through the interaction with Yi-Chui to look at how coronavirus infect the face mask and how we repurpose it, I decided to look at in the electron microscope. So since our lab are not equipped to look at the SARS-CoV-2, we look at this called alpha coronavirus, which is in the family coronavirus, which was discovered almost 20 years ago, and it only causes mild respiratory disease. So here are the cryo-EM images of a single coronavirus. Those I circle out are we call spikes. When you watch evening news, you can always see this circle of a nanoparticle with the red color spike. And in our finding, the spikes are not evenly distributed as you see in your evening news. In fact, the spikes are variable from particle to particle. So once we got the image, we decided to use computer to pick up all the features that correspond to individual spikes. And then we do the class averages. We pick up 82,000 spikes. Each spike is made up of three polypeptides. And so we reconstruct a 3.4 angstrom resolution on the spike proteins. I would say it's not the entire spike, but at least the exterior part of the spike protein represented in 3 different colors. And so in this top view, and this is the side view. And then this the one on the right shows the model of one of these 3 structurally identical polypeptides. Now, in addition to the proteinous material, we see density sticking out, which we interpret as the carbohydrates. So we actually pick up different moiety of the carbohydrates. Before our work last year, 2 years ago, David Wessler over at University of Washington, he actually expressed and purified the spike protein of the same virus. He also used the cryo-EM to look at biochemically purified spike, but not spikes on the virus. And it's interesting enough, both his structure and our structure looks almost identical at these 3.4 angstrom resolutions. But in our case, we actually see more density sticking out because the virus we grow the cells on is different from his. So as a result, the amount of carbohydrates would vary, depends on where the virus are grown. So this is an interesting aspect. Now, we believe that we no longer need to do protein biochemistry. We just look at the pathogens of whatever bioentity, and we can get structure at the atomic resolution. Now, after we get the structure on the alpha virus, we compare those that's been done by others with the COVID-2. Again, those were done based on expressed and purified protein moiety. So they are not done in why the spike still attached to the variant. So we find that there are 5 domains in the exterior in the upper part of the spike in the alpha coronavirus, and there's only 4 domains in the COVID-2. And interesting enough, structurally speaking, the A domain, 0 domain is structurally analogous to the A domain in the COVID-2. And now, but in the other domain B is the business end of this COVID particle, because they bind to the cellular receptor. Our findings shows that the orientations where the receptor binding site is different between the alpha coronavirus and then the COVID-2. So that explains why in the alpha coronavirus, in our case, the NL63, when you bind with the cell receptor, they don't react well at all until we believe that they need another biochemical process to release this receptor binding site before it can bind. And the other interesting analysis of our work is that in this C domain, one of the 4 or 5 domains of coronavirus is both sequence and structurally highly conserved. So this leads us to suggest that perhaps if we want to develop a neutralizing antibody, this is the domain one should target, because you want to have an antibody will kill not only the COVID-2, but all coronavirus from in the entire family. And so this is one approach that we tackle this coronavirus particle. The other approach is we do the tomogram. In other words, instead of taking one single particle images in an area, we can do a tomogram reconstruction. This is a virtual reality display of this tomogram, and all the spikes are there. And you also note that not every particle have the same number of spikes neither. So they're absolutely pleomorphic. So based on this kinds of work, we can actually, from the 3D tomogram, we can computationally pick up the particle, and in such a way that we can do the analysis of the spike. And so what I have shown so far are the spike that has this crown, we call the crown part of the spike, that we are high resolution. But there is another we call the stalk part, which is a long string of polypeptide that penetrate to the membranes of the virus. So we then carry out the we call subtomogram averages on the crown part of the spike, and then another subtomogram average with the stalk part of the spike. And then from there, we can figure out from each of the spikes what are the angle between the crown and the stalk. So as a result of that, we can do an analysis and interpret on all these different averages on all the different spikes. And we discovered there is actually a hinge motion at the upper part of the stalk in such a way that the crown part can rotate. And then for those that are of one conformation of the stalk, then there's a planar rotations in the crown part. So these approaches teach us that this bite that which are the major protein component that cause infectivity are not a static structure. They can actually undergo bending and twisting. So in the context of developing drugs and antibody, I think this kind of information is absolutely helpful. And now I'd like to show you the next example, which have evolved about a chaperone, which I like to show the structural heterogeneity. So far, all the structure I show, we are able to organize the data and process it, give you one single structures, at least to high resolutions. So this is the chaperone, which is help the protein to fold. And it's a protein machine that has 16 identical polypeptides, and under some chemical condition with the nucleotide binding, we call a close conformation. This protein machine has 16 polypeptides. Each color represents one polypeptide. We're able to resolve these at 2.3 angstrom resolution, and this one polypeptide shows how clear it is. You can see the side chain conformations. And in fact, in here, you can see this ATP aluminum fluoride, which is the nucleotide, they can bind to this protein machine. So the questions that we are asking is, in the cell, the ATP concentration can fluctuate, can vary. And so we are interested to look at the property when at a lower concentration ATP under some cellular conditions, what would the structure look like? So we did the next experiment at a lower ATP concentration. Instead of seeing this, all the polypeptide chain close up together, we see these are open. and conformations. And in fact, the resolution and an average is around 4 angstrom rather than a 2.3 angstrom resolution. And if you measure the resolution distribution, each of these protein subunit, they vary with very high resolution close to the equatorial domain and a low resolution and go to the apical regions. So then trigger to our mind that perhaps each of these particles that we see, on average, we can derive an average structure, but in reality, each of these subunits may be different in each of the particles. So we then undertook another data processing step is to classify the protein subunit in each of these particles. So first we break the 2 rings into one single ring, and then we find out by classifying the structural variations, 50% of them do look like group into one group. And in fact, the features in this group densities appear to be well resolved. And then from there, then we use a computer to extract each we consider to be a protein subunit, and then we classify them. We classify in 10 classes. So there are 10 possible conformations from this 50% of data. From there, we can build the atomic model from it because the equatorial and the intermediate domains are good enough for us to build the model. And then from that model, we look back the density map, and we find that some of them has the nucleotide in the intermediate domain and some don't. Now, so these are kinds of analysis that we are now doing, looking at the whole particle. And then we're looking at subcomponent particle event to a single polypeptide. So at the end of the analysis, we have over 1 million subunits of these polypeptides, but they can be classified in 8 different conformations. So the next slide, then so when we combine, and I want to point out, these conformations in red are the most open state without any nucleotide, and this conformation on the right is a more close conformation with the nucleotide, and the other one are something, the structure, something in between. The 4 of them on the right have nucleotide, and 4 of them without the nucleotide. So now when we put all these together, superimposed together, it's quite clear. They are highly identical in the bottom part of this polypeptide, but highly variable in the apical domains of the polypeptide. From here, then we can then, since we know each of these polypeptide, which particle, which subunit it come from, so from there we can actually map back for each particle what the conformation of each of the 16 subunits are, different color denotes different conformations. And from here, then we can build one single atomic model, one particle. Similarly for the second particle and the third particle and the fourth particle and so forth. There are 100,000 of particles. From here, then you can see the distributions of each of these conformations of the subunit that for each particle, they vary from one particle for another. And so from there, we could actually determine the distribution of the nucleotide and also about how many nucleotides per particle. So from here, we then conclude by all these massive analysis of these hundreds of thousands of particles with millions of units within that, then we conclude that the binding of the nucleotide in each of these subunits is a random a stochastic process rather than a cooperative process. So that answers one of the basic biochemistry questions at a single particle level, namely, does the nucleotide bind to this protein complex randomly, or cooperatively. The answer, they are random. So I think now the exciting part of this from the biochemist point of view, we can now address the biochemical questions as one complex at a time. So finally, I'd like to go on my last example, or take only a couple minutes, which is I want to put the study of high resolution into contact with the cell. So we have been working on this called chikungunya virus, which is an insect transmitted virus, and we can purify it. So these are the purified virus embedded in ice. We can generate a near atomic resolution structures to show there are two layers of icosahedrally arranged protein layers. The outer layers are made up of two proteins, and in fact, the protein has a membrane spanning regions across this membrane virus that we in contact with the inner core, which also arranged icosahedrally. So these are all in vitro experiments that do a kind of 3 or 4 angstrom resolutions. Now we start asking the question, can we see these kinds of particles in the cell? So this is an experiment that we do to tomography of the virus infected the human cell. And here is, I'm showing you, here is a tomogram of reconstruction from this chikungunya virus infected human cell. And in here, the really tiny filaments are, we interpret as at the cytoskeleton filament likely to be the actin filament. And then all these one we're seeing are different stages of the budding out, the egress of this virus particle in this tomogram. So by analyzing numbers of tomogram, we were able to classify that some look like a tomato, or a round tomato, which we interpret as the inner nuclear capsids. Some of them are the virus particle partially assembled, some have been more assembled, and some are almost entirely assembled. So I think from this, we can actually, from this sampling from the particle in the tomogram of the chikungunya infected cell, we see a series of different assembly in the mediate. And from that, we can actually map out the number of distributions of what is more mature particle, what is the beginning of particle. We can actually work out some workflow of how these virus particles are assembled. It turned out all of them need to the inner particle to be a nucleusing site to allow the outer capsid protein to assemble. So I think this really open up a interesting approaches looking at the structure in the cell and looking at its assembly process, because in the cell there are hundreds of biological processes. And I think the dream of cell biology is looking at these processes one at a time and under different types of physiological conditions. And in summary, I think I gave you 5 examples. The first example I showed you, within 20 hours data collection, we get an atomic resolution structure. It's become, in my opinion, a very powerful tool for the biochemists as well as the pharmaceutical company who want to develop drug against the protein target. And the other equally exciting case is the RNA, because RNA, as you know, that the vaccine is made up of RNA that many of us have received, and the structural information is going to be really helpful. And so it is a class of molecule not amenable for crystallography for a long time. So I think Cryo-EM become the tool in the future to look at all these RNA structures at different biochemical states. And then now I show you in the alpha coronavirus, we can actually looking the whole intact virus without extracting individual proteins to get to near atomic resolutions. And then the 4th example to show that now we can build atomic model 1 particle at a time, that will really open up a different types of questions that biochemists can answer. And lastly, the last example I showed, the tomography, is really the most promising techniques in the coming decades or that open up the door for cellular and physiological biomedicines. So that with that, all the example, we have been fortunate that 3 years ago been set up by NIH who has the vision that Cryo-EM is the future structural biology. You set up 3 centers. We are one of the 3 centers. We have the missions that to have a state-of-art instrument to open up the global community for data collection, similar to the synchrotron or the femtosecond beam nights in SLAC and other DOE national lab. We also have the responsibility of training the new generations of Cryo-EM users. So I think anyone of you have problems are welcome to come to us because all these are free access, and if you have the appropriate project. And finally, I'd like to acknowledge the people in my group, Kaiming and Jiaming and Greg and Yan, David and Sensen and Mike. They put tremendous amounts of time the last couple of years to produce the data that I share with you today. And this project would not be possible with the very collaborative effort between our lab and Ritu Das in biochemistry, Judy Freeman in biology, Serena Yang in the data science departments in the medical school. And we are also some external collaborator in virology, Jing, and also with the Tomogram average software generated by Mei Yan and Steve Luckett at Beta. Again, thanks very much for your attention.
Well, thank you so much for your great talk. Really beautiful data. Let me ask you a few questions from the audience. The first one is about, can you use the annular bright field imaging to observe the water interaction and hydrogen bonding in cryos?
Okay, so I think we definitely at a stage that at this 1.2, 1.1 angstrom resolution, there are signs of hydrogen atom, the hinge of hydrogen atom. Now the question is whether we could go further about the electronic state of some of these atoms. And something I haven't had time to talk about is the density features between neutral atom, positive atom, and negative atoms. My answer is yes, definitely that is the case. Particularly the protein have some negatively charged amino acids, and those detections can be more apparent at lower resolution, not necessarily high resolution. So this is one of the really active research we are trying to understand the physics of this scattering between different types of ionic states of the atoms and also different atomic number. This is a good question. This is under very serious studies.
Hey, wow. So another question, how do you distinguish the magnesium density from the noise density?
Okay, very good question. So in the paper we published last year, we actually use a method we call a SWIM method, okay, which we rely about the locations of the signal, which is away from the biological protein or RNA. And then the distances, because there are biochemical rules. What are the expected distances between the water and the oxygen and the nitrogen atoms in proteins, and also same as the ions. So we actually use the same rule as set up by crystallography to look at the distances. And number one, and number two, we look at the amount of signal about the surrounding density, at least the 2 sigma around the surrounding density. And the third criterion we use, I mentioned very briefly about, we call the Q score. In other words, those putative water molecule or the ions molecule, the score has to be comparable to what is expected in the analysis we did from the structure that deposit in the protein data bank.
Yeah. Next question was, for your spike protein, how big is the hinge region? What kind of amino acids are present in the hinge region of spike protein?
Okay, so that's a good question. One thing I did not mention yet in detail is about the carbohydrate part. One of the concerns we have, because throughout the spike protein, there are lots of decorative glycans, and that's after our 3.4 enzyme structure analysis, we actually decide to go to a mass spec lab in that determine what carbohydrates in our coronavirus. And so I think so we now know the carbohydrate part, and in addition, we actually engage with a group who did molecular dynamics studies. So now we have done quite extensive molecular dynamics with a group in Arizona State University, and then we looking at the energy landscapes of these kinds of rotations and twisting motions. And now that what we observe experimentally seem to be consistent what expected in the molecular dynamic simulation. So these are very exciting. And so there are some of these amino acids in the stalk part is actually they form a coil helix. One thing I did not mention is that in our subtomogram average, this stalk part, the alpha helix is just about to be resolved, but not quite, around 10 angstrom in that pop-up. And so I think so that's a lot of possibility in further along with that. And we are also trying to increase our database, our data set, so that we can get high resolutions.
So while I ask you a last question before passing it to Jen, what's the top 3 things in your mind related to Cryo-EM technique development down the road for a decade to come?
Okay, that's a good question. I think one of them, I'm really keen about this heterogeneity problem, because I think that all the macromolecules, they are not static. They are undergo a continued dynamic motion. So one of the niche in Cryo-EM is able to contain those informations. Now, we are very, very powerful tool like Reliant and CryoSpark and CryoDragon. So I think one of the future area is to develop using the AI-based, artificial intelligence-based algorithm to explore the analysis of our types of data. So we would be giving not only a few discrete confirmation, may probably give you a multiple or somewhat continuous confirmations. And I think combine that with molecular dynamic simulation, I think this would be a good area in exploring on the single particle biochemistry of macromolecule. The other most important and exciting thing is look at tomography. I show you a very quick glimpse. In fact, we have multiple projects in the lab now is looking at the molecule inside the cell. Now, I show you an example looking at very large virus particle. I think we have now projects that are pushing to a kind of sub-nanometer resolution, looking at the cytoskeleton proteins and molecular machines under different types of conditions. I think those, in my opinion, rely very heavily on data processing. And lastly is if we're looking at biology as a whole, we probably require more than one Cryo-EM. We want to link it with fluorescent microscope. So I think the future is looking at the fluorescent signal of the target, the molecule, on one hand, and at the same time looking at the same molecule in electron tomography. So I think that integrations also coupled with maybe before the fluorescent microscope, there are other physiological measurements. We can link the physiological measurement with fluorescent microscope, with tomography, with image processing, so we can account across length scale analysis. And I think similar study can be applied to material, non-biological material as well.
Well, thank you for your beautiful talk and the amazing insights. With that, I'll pass it on to Jen.
Awesome. Thanks so much, Yi, and thanks, Wa, for an incredible talk and for highlighting what you view as the next decade of exciting research in electron microscopy. As many of you know, in our EMX Symposium, we often like to focus one speaker who is more targeted on bio applications and another speaker who's more targeted to energy applications or the physical sciences. And it is my pleasure to introduce the next and also the last speaker of our EMX Symposium this year, Professor Lena Kourkoutis. I had the pleasure of meeting Lena, I think about 5 years ago at one of my first electron microscopy workshops at Berkeley. There she gave an absolutely phenomenal talk on the work that she had been doing at the time using Cryo-EM for battery materials and energy storage materials. Just a little bit of background on Lena. She is a professor at Cornell University, where she also serves as a director of the undergraduate program. She received her diploma in physics from the University of Rostock, Germany, and then moved to Ithaca, where she was awarded a PhD. She then took a short hiatus between her PhD and returning to Cornell as a professor to be a Humboldt Research Fellow at the Max Planck Institute, where she really got a chance to dive into cryo-electron microscopy. And for her research, she's been awarded a number of prestigious fellowships and honors, including, for example, the Packard Fellowship for Science, the National Academy of Sciences Kavli Fellowship, the NSF Early Career Award, and the Presidential Early Career Award for Scientists and Engineers. So, Lena, it's my pleasure to hand over the floor to you. Thank you so much for joining us today.
Excellent. Thank you so much. And I think I'm sharing my screen now. Is that right?
Yep, perfect.
Okay, excellent. So this is wonderful. Thank you for everyone who's sticking around for this part, for the last talk of this symposium. Hopefully you will enjoy this last part as well. And we're going to move from biology to quantum materials. And it seems like a big leap, but what I want to show you today is that cryo-electron microscopy can also provide actually extremely important information about quantum phases at low temperature. So as you heard, my introduction to a cryo-electron microscopy was in a biolab at the Max Planck Institute in Martinsried with Baumeister. And there I really learned about the tools that biologists have developed in the field. And what I want to show you today is that for quantum materials, we can use some of those tools, but there are new tools that are required. And the motivation for cryo-electron microscopy in this field is also quite different, right? In biology, it is to stabilize a phase that is hydrated, that you can arrest at a certain temperature. In quantum phases, what we really want to be able to do is we want to get to a state that only exists at those low temperatures. And ideally, right at the very end of the talk, I want to show you what we really are after is we want to be able to tune temperature and learn how a state emerges. And what I thought was really interesting, that why as one of the kind of frontiers in cryo-electron microscopy was to think about heterogeneity. And heterogeneity is, of course, very important in biology. It's also very important in quantum materials. And what you see here on the first slide is an electron micrograph of a quantum phase. And what you see directly in real space is the heterogeneity that exists in these quantum phases that can be resolved with extremely high spatial resolution using electron microscopy. And to just put things into perspective, the little arrows that I color-coded here are actually displacement of single atomic columns by just a few picometers. And those atomic scale displacements or picometer scale displacements give rise to a new electronic phase. So the first talk today was all focused on TEM. And so what I want to do first is switch my slide. And I can't switch my slide. There we go. Okay, excellent. That would have been tragic. So you heard about TEM, where you have a parallel beam that illuminates the sample. Here, this is a little bit different. Here we focus a very, very small electron beam onto the sample. The electron then interacts with the sample, and you can record a number of different signals. The most straightforward is to just look at the elastically scattered signal, and that produces a two-dimensional image when you raster the beam across your specimen. And so here you see an interface between two oxides. You see the atomic scale resolution you get from that simple interface. And just by looking at this interface, you see the new reconstruction that emerge as you join these two materials together. Now, this is the simplest way you can measure. You can imagine replacing this detector with a two-dimensional pixelated detector. And I will not go into the detail, but I want to just highlight recent results from my colleague, David Muller here at Cornell, who has used electron ptychography to push the resolution of electron microscopy further down to 0.2 angstrom resolution using electron ptychography, which makes use of all the scattered electrons of your electron probe in the STEM mode. And so if you want to learn more, feel free to look at this recent publication. Now, in addition to the elastically scattered electrons, you can also look at the inelastically scattered electrons. And I took a look who spoke at the last symposium. And if you were there, it was exactly focused on this, which is ELS, electron energy loss spectroscopy, to study the chemical as well as electronic states in a material. And I just want to highlight one example here again. It's an interface between two oxides. And what we have been able to do is not only map the distribution of elements in that sample, but we have also been able to map the distribution of charge right at this interface. So here you see that we have about 1.1 electrons at its peak accumulating at that interface. And those kinds of electronic reconstructions are important to stabilize an electronic phase at an interface between two materials. More recently, we have used electron energy loss spectroscopy to also look at the electronic structure of nickelates, which I don't want to spend too much time on. This is a class of, actually a new class of superconductors that was recently discovered, in fact, at Stanford by the group of Harold Wang. And it's beautiful work that shows you the power of electron microscopy to study electronic states. Now, today, in today's talk, I want to focus on cryo-electron microscopy, right? Keep this in mind. This is what we want to do. This is not what we can do at the moment. I want to go back to imaging at very high spatial resolution. But let's look at the nickelates. And here, I'm plotting, in fact, I'm plotting two physical property measurements of this material. This is the bulk phase, the 113 phase, and this is the superconducting phase. And all I want to take you for you to kind of see from this is that here you have a material that goes through a phase transition. And that phase transition, this is actually metal to insulator transition, happens well below room temperature. In fact, it happens around here 180 or so Kelvin. Here on the right side, you actually see the conductivity, and it drops down to here. This is where the material actually becomes superconducting in the reduced phase. Now, the fundamental problem with our measurements is that most of our measurements have been done at room temperature. And of course, we are here today to use cryo-electron microscopy to access these low temperature phases. And for now, I want to focus on phases that we can access using liquid nitrogen cooling, similar to the cooling that what you described a minute ago. You will also see that in order to reach the superconducting phase, we need to go to lower temperature. Okay. So this is, of course, not the only material. Here is a phase diagram of a manganite. And what you see is that a lot of the interesting physics actually emerges at low temperature. And so this is kind of from my introduction that I mentioned earlier. Cryo-electron microscopy in quantum systems, really the motivation is that the phases exist only at that temperature. So we really need to be able to run our instruments as well as our experiments at the relevant temperatures. And ideally, we want to be able to track how things evolve as you go through a phase transition. Now, why electron microscopy? Quantum materials have been studied with many probes before, including at liquid nitrogen temperature and even liquid helium at sub-liquid helium temperature. But the advantage of STEM in particular is that we can probe the atomic scale structure and electronic structure. We can probe it within the material, not only on the surface. And we can map it both locally and over large length scales. And that lets us access the heterogeneity that I introduced to you in the beginning, which is to understand the role of defects, the role of spatial correlations in a quantum system. And often these correlation or competition between two phases gives rise to these new quantum phenomena, or why these materials act differently than others. So this is the motivation. It seems straightforward, especially since we heard Watt talk about all the beautiful things you can do in cryo-electron microscopy in biology. Now, the problem is it's not that simple and that does not mean it's simple in biology. It's absolutely not simple. But why can't we run all of the techniques that I've described so far for crystalline materials at cryogenic temperature? And here's the fundamental issue. So for quantum materials, typically what we do is we use site entry holders where your cryogen is held here in this tiny dewar, the sample sits at the front end. And so when you then measure over time, then this is representing directly the problem that exists in the experiment, which is rapid drift in this of the sample over time. And so here we are measuring or quantifying that drift, and you see that the drift is about 1 unit cell every 2.5 seconds. And so this is a serious issue, especially if you want to map, for example, the electronic structure across an interface, which requires longer acquisition times than imaging. So fundamentally, the stability of these holders is limited. Now, this is a standard site entry cryo holder that is commercially available today, and it's operating. We have assessed many of them. This is using a Titan-based microscope. And you see that the drift rate is on the order of 0.75 angstroms per second. So this is our limit. This is what we have to overcome. Now, let me show you what the drift rate will look like in one of Watt's microscopes. So here is a Thales Arctica. It has a dedicated cryo stage, not a site entry holder. And you see this is plotted on the same scale and all of them, all of the over the same period. And you see it's condensed into a point. And that is because the mean drift here is much, much, much lower, an order of magnitude lower. And so this is extremely promising because it means that we have high stability that may allow us to do spectroscopy on these systems. Now, what is the problem? Why are we not using? In fact, this is our Thales Arctica here at Cornell. Well, the problem with this instrument is that it lacks dual axis tilt capabilities. So in a crystal, compared to a biological molecule, we need to be able to orient our crystal onto a crystallographic zone axis. And that means that we need two tilts or a tilt and rotation, or any combination. And that currently does not exist in biological dedicated cryo instruments. And so we are currently limited by the drift with our site entry holders. So how, for imaging, we have overcome this limitation. And the way we are doing it is similar to what is done in biology, which is you record basically movies of your image or of your region of interest. And what we then do is we set up a registration method that is optimized for extremely low or low signal to noise data. And that allows us to get to the signal to noise ratio that is needed for highly precise measurements of the atomic crystal. Okay, so here you see just one example. These are actually quite still high signal to noise images, but you see that registration of an image series allows you to push up the signal to noise and resolution down to close to 0.6 angstrom, as you can see here in the Fourier transform. So what we can do today in real space without any averaging over different areas in the sample, in a single area of the specimen, we can image the structure of the material at sub-angstrom resolution. And what this shows in this particular material is this is 1T prime tantalum disulfide, and it actually shows how it trimerizes as you cool it down into the electronic phase. I don't want to talk too much about this particular system. What I want to do instead now is I want to show and talk a little bit about one example of really the importance of understanding heterogeneity in quantum system using charge-ordered systems as an example. And so if you think about charge-ordered system, charge density wave system, often what these kinds of systems include is that you start with an ideal lattice, and that often the lattice modulates or distorts in this charge-ordered phase. And what we can now do with cryo-electron microscopy, we can probe this modulated lattice in real space. Now, these modulations and these charge-ordered states are important because they correlate with the electronic properties of the material. Here, for example, you can transition from a metallic state to an insulating state to create a switchable material. And that switchable material can be tuned by temperature, pressure, as well as doping. Charge ordering has been very important in the last, or has gained interest again. It has been widely studied a long time ago, and it's now interesting again because there is, of course, a close connectivity with superconductivity, where it competes with superconductivity. It has also been shown to play a role in establishing topological insulators. And so there's a lot of interest in understanding these types of phases. And what we will be using for now is we're going to be using the lattice as a metric to probe our charge-ordered phase. Now, as I mentioned before, electron microscopy has been used for these charge-ordered systems before. So this is a charge-ordered manganite that was studied in the late 90s. First in diffraction, and in diffraction, what you'll see is superlattice peaks that decorate the Bragg spots of the lattice. And these superlattice peaks can be used to image the ordered phase in our manganite system. And these are the famous stripes in the stripe-ordered manganites that were imaged using dark field TEM in the 90s. But what we want to understand is we want to understand precisely how the atomic lattice distorts within that charge-ordered phase in order to understand the interaction, and then ultimately design a material that acts in a way in order to, for example, tune the transition temperature or the response or the tunability of that material. Okay, so the goal is to map within one of these little domains. So we started with a prototypical manganite. It's the half-doped system. In fact, also grown at Stanford by Harold Wang in thin film form. And what you see here, at low temperature, you are sitting in this charge-ordered phase. Now, by straight imaging in the cryo stem, if you take the Fourier transform, you can again see these superlattice structures that arise because of that symmetry breaking when you're on the charge-ordered phase. So we know that we are indeed sitting in that phase, and we could do dark field TEM to image our stripes or whatever structure we have in this system. But what we want to do is we do not want to just image in dark field. We want to actually map the picometer scale displacement of the atoms. And the reason why we would do this is that we want to understand the nature of this charge ordering. So charge ordering has been studied for many years. There are two primary models. One that you see here, plotted in 1D for simplicity, is the site-centered order, where charges localize on an atomic site. And there's a competing model where the charges are in the bonds, as you can see here, indicated by this blue circle. Now, this is the 1D simple model to just present the general idea. Of course, the 2D picture might be more familiar to you. So here again is the site-centered order. You again see the variation between the manganese valence, where you have charge disproportionation between the individual sites, versus the bond-centered order, where this is not the case and the ordering occurs on the bonds. Now, this is the typical electronic picture that you see in literature, these orbital ordered structures that you see. But there is what you should not forget is that there's an atomic lattice picture. And that means that the atomic lattice can respond and can distort. And so here, what I've now done is to color code the individual manganese sites. And you see that in this particular site-centered symmetry phase, the first manganese atom will displace downwards, the second will be stationary, the next one will go up, 0, down. So we have a down, 0, up, 0, down movement. Okay, this is the site-centered symmetry. And if you go to bond-centered symmetry, the distortion and the displacement pattern changes, where you have now 2 down, down, up, up movements. So we can use this picture of charge ordering to track in real space the heterogeneity and competition between these individual phases. Okay, so skipping lots of steps, we can now look at the results at near liquid nitrogen temperature. So this is the manganite crystal, it's a thin film image at low temperature, and we are now tracking the displacement of the atomic columns at low temperature. Each blue arrow here is an atomic column, the manganese site, displacing upwards by about, let's say, 7 picometers. And so the picture that that arises is that you have an up 0, down 0 movement of the atomic columns. And that, of course, will remind you immediately of the site-centered symmetry. So what this shows is, using real space measurements, we can now track directly the symmetry, the local lattice symmetry in this electronic phase in the material. So this is actually a model that has been is well understood from the 60s, and now we provide this real space picture of the site-centered symmetry. Now, that itself would be interesting, but what's more interesting that if we go to a different part of the sample, that we see an entirely different symmetry. And so here you see an up, up, down, down, up, up movement. And that pattern will remind you of the bond-centered order, with one small caveat, which is that the displacements here should be the same versus in the experiment, where you have clearly one side that is stronger displaced than the next side. And that brings us to a predicted intermediate phase, the intermediate ordered phase that has different symmetry, which now is distinct from bond-centered order, with a displacement pattern that is down, down, up, up. However, the amplitude of the modulation is different in the 2 atomic sites. So what we have discovered using cryo-electron microscopy in real space is that there's also areas in the film where this intermediate order is stabilized. So if you look at both, then we have site-centered symmetry in parts of the sample and intermediate order in others. And this is important because this is non-centrosymmetric, so you can start thinking of coupling to this in the future. Now, what's more interesting, and that brings us to heterogeneity, is we can now zoom out in a region of the film. And so if you zoom out, then what you will see that you have a transition from the top left to the bottom right, where you go from site-centered symmetry to intermediate order on the bottom right, with a transition region in between. And so what this shows you is that oftentimes in quantum system, you have coexistence of ordered phases, and they coexist on the nanometer scale. And often bulk probes will not be able to get at those kinds of heterogeneities that exist in real space. So CryoStem really is a powerful probe to measure both local symmetries, but also variations and phase competition. Okay, so I showed you the half-ordered system. We can also do this in a 2/3 system where the periodicity changes. The main reason for me to show this very briefly here is to show you that here, again, heterogeneity plays a very important role, where phases, the two different colors that you see here, the red-green and the blue-yellow, are competing phases. And you again see areas where when one phase arises, there's disorder. The second one, if you look carefully at the yellow stripes, you'll see that they actually distort. Okay, so this is where we are at liquid nitrogen temperature. And I think there's a lot of low-hanging fruits where phases exist of interesting quantum systems that we can now access. I just plotted here, again, the prototypical manganite phase diagram. Now, what we really want to think about, though, is what if there are phases that are stable in narrow temperature windows? Or what happens as you go into or across a phase transition? And for that, we cannot be static anymore. We need to be able to tune the temperature. And so what I now want to show you here is that we have also been able to obtain stable sub-angstrom resolution imaging at all temperatures between about 100 Kelvin and then all the way to 1000 Kelvin if you choose, all done in one cryo setup. And that now allows us to really probe transitions and how the atomic lattice responds in real space. Okay, so how do we do this? So let me walk you through it. On the left is an electron microscope. You again see a site entry holder right here, but this site entry holder is different than the standard Dewar models that are attached here to the rod. But instead here we have braids that hang into this very large 40-meter Dewar. This holder was designed by Henny Zandbergen. But what's so important about this design is that at the tip of the holder, it includes a small MEMS chip. And the MEMS chip allows you to place your sample on it and to locally heat the sample, which then allows you to tune the temperature of the sample while the entire holder sits at liquid nitrogen temperature. Okay, so this is quite important because it allows us to change the temperature very quickly, maintain atomic resolution, and we basically avoid introducing a lot of drift. Let me show you one example. So here's an example that is a family of 2D cluster magnets, niobium halides. Like other 2D magnetic materials, such as these famous, there are quite a lot of new interesting 2D magnets that we would like to go after. However, this particular material is convenient because the transitions will happen at relevant temperature. Here, in this particular material, it's actually above room temperature. But we'll show you in a minute why that's also important for cryo-electron microscopy. So these are structurally different transitions. And what I want to now know is, well, how does this transition occur? So here is the high temperature phase. You notice already I'm not at room temperature because the entire setup is liquid nitrogen and I heat to the high temperature phase. I could go to room temperature or whatever I want. But this is the high temperature phase, and you see the characteristic structure when you look in plan view, which is that we are now looking at it top down on the structure. When you cool the material down, we are now sitting at 223 Kelvin in the same experiment. You see that you have a very distinct structural transition, and that is reflective of a change to a 6-unit cell repeat compared to the 2-unit cell repeat at the high temperature phase. So yes, we can cool into the low temperature phase and resolve the structure. Now, what's more important, we can do this at intermediate temperature and continuously. And what we actually see, I'm only showing one state, that you have intermediate phases that are stable as you cool or heat. So here you see another phase that is very different than all other phases that we have observed. And then as you continuously heat, you then recover again the high temperature phase. So these are actually just 2 intermediate temperature. We have many, many more to show. And what this result showed, not only that we have to actually, so first of all, it shows that in these thin films, that the hysteresis becomes very large. You see that we have to go all the way to 670 Kelvin to recover the high temperature state. But what you also see, or what I hope you believe me, is that there are many, many intermediate states that are stable. And that is quite interesting because it might suggest that there are intermediate magnetic states in these systems as well. And that might give rise to these broadened distributions in the hysteresis loops of bulk measurements. So I have 2 minutes left. So let me summarize what are the prospects and where are we today in quantum materials, and in particular on cryo-electron microscopy for quantum materials. So first of all, I showed you that for quantum systems, you need to just, many of them, the phases of interest emerge at low temperature. Therefore, we need to go to low temperature to, in fact, access them. The temperature at which you want to work depends on the material system. There are lots of interesting systems at liquid nitrogen. There are many more at lower temperature. And there are lots in intermediate temperature. With our setup, we can map both local structure, you see one example here, local symmetries of the crystal. But what's more important, we not only can measure local symmetries, but also how these local symmetries vary over nanometer length scales. We can look at heterogeneities, phase competition, defects, all of the things that make quantum materials work the way it does. Where are we currently in terms of cryostem measurements? I've showed you that we can both measure picometer scale or sub-angstrom resolution, picometer scale precision at both liquid nitrogen temperature and intermediate temperature through MEMS heating. I have not talked about spectroscopy where we are there, so we can map the elemental distribution at low temperature. But really what we are after is measuring electronic states. So can I map the distribution of electrons now here, not just the lattice? And that requires new advances in instrumentation. So what we need in terms of instrumentation is, number one, we need much more stable cryogenic operation in order to not only map electronic lattice displacement, but also the electronic structure in these systems. And we need to extend the temperature range at which we can access quantum materials. And that means since we have covered liquid nitrogen to room temperature and up, we want to also push down in temperature. So here is kind of slide 2 or 3 from my talk, which was the nickelate superconducting nickelate that I showed earlier. Right, working at liquid nitrogen temperature has allowed us to access the metal insulator transition in the nickelates, or will allow us to access this. But by going to, so first of all, we can measure this continuously and therefore track how these evolutions happen. This is what we're after. And then, of course, we want to push down and actually measure in the superconducting state of the material. So with that, I want to thank the people that make this work. And that's both my group. In particular, I want to highlight Ismail, who's not on this picture. He is now at Harvard. Barrett, who's right here, and Elizabeth, who is here. And they all have masks on, so sometimes a little bit more difficult to recognize. But I also want to highlight all of my collaborators who really provide such excellent materials and who are active, always active parts of this collaboration. It's been a lot of fun working with many, including Harold at Stanford. And so with that, I want to invite you all to our summer school, which happens to happen next week. And there's one advantage of COVID, which is we have to unfortunately run it virtually. And so with the virtual environment, we are going to open up the lectures to live stream. And so please join us if you're interested. You're going to hear about STEM, cryo-STEM, as well as tychography. And yes, with that, I'm happy to take questions.
Lena, thank you so much for an outstanding presentation. This was incredibly exciting and really challenging work. I am so impressed at the resolution you've gotten and at the phases you can look at. There are some questions in the chat and in the Q&A. I wanted to kickstart the Q&A by asking, when you are kind of correcting for some of the drift or the motion, do you treat the entire material as a rigid object, or are you also correcting for kind of local motion at the atomic level as well?
Yeah, so there's different camps, and some use non-rigid registration, which accounts for local variations. We do not do that, I'm always, my belief is you treat the data, you massage the data as little as possible. So my worry is always that you modify the results in this, and so we try to avoid it. Ours are rigid. And for those of you who do rigid registration, there's now, including Thermo Fisher, they have all these automated routines. I will tell you that they don't work. I mean, be careful. If you have very high signal to noise, that works. For our systems, these charge-density wave systems, where you have a single crystal that's almost perfect, you often get these unit cell hops. And if you get unit cell hops, your quantum state will wash out as you average. And we have shown that using some of the routines that are available on microscopes. So, feel free to use what we have done or come up with your own method, but just be careful when you do averaging. I mean, my hope is at some point we don't have to average, but at the moment we don't have the stability.
Cool. Yeah, thanks so much for that answer. Another question is, you mentioned looking at electron energy loss spectroscopy to try to unravel what the electronic structure is. Do you think, and I can imagine, or I know firsthand how challenging that is to do, especially when there's, even small bits of drift, and you're trying to understand what the atomic constituents are doing or what the electrons are doing. Do you think there's an opportunity to also do something like, holography, or something where you can kind of probe structural changes to the magnetic states, not just looking at the electronic structure, but the...
Yeah. So this is actually funny because I happen to have this open. So this is work by my colleague David Muller, who is looking at magnetic skyrmions, and has actually gotten quite high resolution magnetic imaging. And the way he does that is using electron tachography to image the magnetic structure in the material. And so you happen to also see that we're working here at low temperature. So yes, combining low temperature with magnetic imaging through tachography is definitely promising. And tachography, of course, has other advantages of being able to track electronic distribution as well, but you have to be a little careful there because you're dominated by the core electrons. And so if you're interested in kind of the electrons that are really doing the work, those signals are small. And so we believe that we can get there with EOS, we have a new microscope on campus. Barrett actually ran the first cryo-electron EOS experiment yesterday after sign-off. So we hope to be able to show it soon, but I think we need better stages to get there.
Cool, thank you. Well, I'm also a big supporter, or if there are better stages, I will love to use them. There are a few questions in the chat, but just since we're close to the top of the hour, I'll end with a final question that kind of parallels the question Yi had for Hua. In addition to better stages, what do you think is the future of electron microscopy for quantum materials in the next 10 years? Like, what do you see as the most exciting potential breakthroughs?
I think, well, stable, we should not underestimate how much work it is I've been trying to get stable stages realized with different companies and so on for a number of years now. So there's a big hurdle to overcome, and I think that should be a priority. The other one is, of course, to extend the temperature range. As I mentioned before, we should invest in that. The experiments I showed you today on intermediate temperature are done on it's a commercial system. However, it's not, it's still not extremely user-friendly. It allows us to look at those states. So we want to be able to tune states more. And that includes both temperature control It also includes biasing control within the microscope. It includes other tuning knobs that allow us to really look at, well, how does the charge density wave glide in the material, for example, as you excite the material. So all of that has to be done at low temperature. And so I think we need to work on controlling the sample at those relevant temperature and remain stable so that we can get at the relevant physics, which is smaller length scales and smaller energy scales compared to other material systems.
Cool, super exciting. Thank you so much, Lena, for an outstanding talk. And Bob, I will hand it back to you.
Thanks so much, Jen. And thank you both, Wah and Lena, for your fantastic talks and also for addressing the questions about where we're going to go in the future, which is electron microscopy seems to be just getting better and better and more challenging. And so there is so much work to be done and derive fantastic information like you both described about your respective systems today. So really fantastic. We will stay online for people who want to join us, but we will be closing fairly shortly. I'd like to thank the people also who have helped get this going. I mentioned Ziwen and Raphael, but also, of course, Yi and Jen and Wah have been so enthusiastic about this. And we think that this has been a good exercise to keep our community together when we can't meet physically. And we do it by the airwaves, as it were. And we will, it is our full intention to resume again in autumn, as I mentioned, October 4th, October. But with that, I hope that everyone in the meantime uses the summer as a period of rejuvenation. Perhaps we will all be back at normal, regular work fairly soon. I'd like to wish everyone that good health, and we'll see everyone in the autumn when we start again. And why not drop us a line? And if there's anyone that you'd like to see, we have so many speakers lined up already who are going to be equally exciting and different in many ways. But if there's anyone that you'd like to hear us put into the program, just send us a note, an email to any of us independently, and we'll see what we can do. But thanks again, Wah and Lena, today. Fantastic. And see everyone in the autumn. Thank you.